Welcome to this lecture on statistical errors, specifically focusing on type 1 and type 2 errors. In the realm of computational biology and zoology, we constantly analyze massive amounts of experimental data to make scientific discoveries. However, data is inherently noisy, and our statistical tests are never absolutely perfect. Understanding the types of mistakes we can make is the absolute foundation of making safe, reliable, and impactful scientific discoveries. Let us dive in and explore how we categorize and quantify these unavoidable risks. To begin our journey, imagine you are a doctor using a brand new diagnostic test to check if a patient has a rare disease. When you run this test, there are two ways it can completely mislead you. First, the test might scream that the disease is present when the patient is actually perfectly healthy. This is a frustrating false alarm. Second, the test might say the patient is healthy when they actually have the disease, meaning you missed the danger entirely. In the world of statistics and computational biology, we deal with these exact same mistakes every time we analyze experimental data. Now that we understand the basic intuition of false alarms and missed dangers, we need to formalize our scientific questions. To formally define statistical errors, we first need to understand the concept of a null hypothesis, denoted by the symbol H0 in statistics. The null hypothesis is the boring default assumption, such as claiming that a drug does nothing or a patient is healthy. On the other hand, the alternative hypothesis, denoted by the symbol HA, is the exciting discovery we are actually trying to prove, like claiming that a new drug cures the disease. Every statistical test is a battle between these two hypotheses. With our hypotheses clearly defined, let us look at how our decisions can go wrong. A type 1 error occurs when we incorrectly reject a true null hypothesis. In plain terms, this means we claim a scientific discovery that is not actually real. It is our false alarm. Conversely, a type 2 error occurs when we fail to reject a false null hypothesis. This means we miss a real biological effect that was right in front of us, our missed danger. Imagine a pharmaceutical company testing a cancer drug. A type 1 error approves a useless drug, wasting billions, while a type 2 error abandons a genuine cure. Now that we know what a type 1 error is conceptually, let us see how we represent it mathematically. This formula defines the probability of committing a type 1 error, which we denote with the Greek letter alpha. Conceptually, this comes from conditional probability. The notation P stands for probability, and the vertical bar means given that. So this equation translates to plain English as the probability of rejecting the null hypothesis given that the null hypothesis is actually true. Alpha is also known as the significance level of our test. If we decrease alpha, we make our test stricter, which reduces false alarms, but unfortunately increases the chance of missing a real effect. Before we move on to the next error type, let us see this with a concrete example. Suppose a clinic is evaluating a new rapid test for a dangerous virus. We are given that the null hypothesis is that the patient does not have the virus, and the alternative is that they do. The manufacturer states there is a 5% chance of a false positive. Why does this matter? A false positive means the test says the virus is present when it is not. This perfectly matches our definition of rejecting a true null hypothesis. Therefore, our alpha is 0.05. Physically, this means out of 100 perfectly healthy people, 5 will be incorrectly told they are sick, leading to unnecessary quarantine. Having calculated our false alarm rate, we must now look at the mathematical representation of our missed dangers. Here, the Greek letter beta represents the probability of committing a type 2 error. The formula calculates the probability that we fail to reject the null hypothesis, given that the alternative hypothesis is actually the true state of reality. In plain words, the disease is there, but our test missed it. The complement of this error, calculated as 1 minus beta, is called the power of the test. Power is the probability of correctly finding a real biological effect. If we increase the sample size of our experiment, we can decrease beta and increase the power, making our test much more sensitive. Now that we have defined beta, let us return to our rapid viral test example to find its value. We are given that the test has a 10% chance of giving a false negative. We need to map this real-world term to our statistical definitions. A false negative means the test says the virus is absent when it is actually present in the patient's body. This perfectly matches the definition of failing to reject a false null hypothesis. Therefore, our beta is 0.10. In physical terms, out of 100 people who genuinely have the virus, 10 will be incorrectly told they are completely healthy, allowing them to unknowingly spread the disease. We have seen how errors work for a single diagnostic test. But what happens when we scale up? In modern computational biology, we rarely test just one hypothesis. Imagine a biologist analyzing a DNA microarray to see which of 10,000 genes are mutated in a specific cancer cell. We are now testing 10,000 independent null hypotheses simultaneously. Even if we set a very strict, low probability for a type 1 error on a single gene, running thousands of tests means those small probabilities add up. This massive accumulation of false alarms is a famous issue called the multiple testing problem, and it is a major hurdle in genomics. To understand exactly how severe this multiple testing problem is, we need a formula to calculate the damage. This equation comes from the concept of expected value in probability theory. It calculates the raw number of mistakes we expect to make just due to random statistical noise. The variable n represents the total number of independent hypotheses, or genes, being tested. The symbol alpha is our chosen significance level for each individual test. By simply multiplying n by alpha, we find the expected number of false discoveries. If n increases, the number of false discoveries increases proportionally, showing how small errors accumulate rapidly in large-scale computational biology. Let us apply this expected value formula to our DNA microarray scenario. We are given that a computational biologist is testing 10,000 genes, so n equals 10,000. The researcher sets the type 1 error rate, alpha, at 0.01 for each individual gene test. We are asked to find how many false discoveries will occur by pure chance if absolutely none of the genes are actually mutated. We multiply 10,000 by 0.01. The final answer is 100 expected false discoveries. Physically, this means even if the cancer cell is completely identical to a healthy cell, the computer will falsely flag 100 genes as being mutated. This is why advanced statistical corrections are mandatory. To consolidate everything we have learned, let us look at this decision matrix, often called a confusion matrix. Describe what you see. The columns represent the actual hidden reality of biology, whether the null hypothesis is true or false. The rows represent the decision our statistical test makes. If reality says the null hypothesis is true, but we reject it, we land in the top left box, a type 1 error, or false positive, governed by alpha. If reality says the null hypothesis is false, but we fail to reject it, we land in the bottom right box, a type 2 error, or false negative, governed by beta. The green boxes represent the correct scientific conclusions we strive for. We have reached the end of our topic, so let us summarize the key takeaways. First, statistical errors are the unavoidable risks we take when making scientific decisions based on noisy experimental data. Second, a type 1 error, measured by alpha, is a false positive, where we claim a discovery that does not actually exist. Third, a type 2 error, measured by beta, is a false negative, where we miss a genuine biological discovery. Finally, as we saw with the multiple testing problem, large-scale computational biology requires careful experimental design and statistical power to minimize these errors. You now have the foundation to deeply trust and evaluate computational biology results.